This is an A-level biology presentation looking at stem cells. In this presentation, we're going to look at the definition of a stem cell. We're going to look at the different types of stem cell. We're going to look at how specifically pluripotent stem cells can be produced. We're going to, of course, talk about the use of stem cells in terms of human disorders and do some evaluation. To get started, make from memory a list of the types of mutations and the causes of mutations in the form of mutagens. There are 6 main types of mutations and there are 5 main mutagens you need to be aware of. Pause the presentation, come up with as many of them as you possibly can, and when you're ready you can restart the presentation. So pause the presentation now. If you've gone through the other presentation on mutations, then you should be familiar with these images, and the 6 types of mutations are addition. It's in old terms would have been referred to as an insertion, but you need to use the term addition. We have a delete mutation. We have what's called an inversion mutation, where we flip the sequence around. We have a substitution. We have a duplication, and of course we have a translocation mutation. And you need to be aware of these names and what each one means in terms of the changes that will actually occur. In terms of mutagens, there are 5 main types of mutagens. We have our ionizing radiation, alpha, beta, and gamma. We have smoking, we have carcinogenic chemicals, ultraviolet light, and of course X-rays. When people hear the term stem cell, they think of only one particular source of stem cells. There are actually several sources of stem cells. Each of the pictures in front of you now represents one of those sources. Pause the presentation, try and work out what the 4 pictures represent, and when you're ready, you can restart the presentation. So pause now. The 4 main types of stem cell sources are embryonic stem cells is the one that most people are aware of. It is also possible to pick up stem cells from an umbilical cord. We can also get stem cells from a placenta, again normally taken straight after a birth. We're also able to take stem cells from adults. We don't actually have to have embryos, fetuses, or placentas at all. We can actually take them from healthy adults, and there's a lot more research that's occurring in recent years that's finding there's a lot more types of adult stem cells than we originally believed. In terms of the types of stem cells, there are 4 real levels of differentiation, and stem cells are grouped based on their ability to differentiate. Remember, differentiate is where we have a cell that is given a particular role by turning on or off certain sections of DNA. When we differentiate, we then become specialized cell, and that's a cell adapted to a particular function. Stem cells are cells that have not adapted to a particular function. They are undifferentiated, they are unspecialized. When we have a stem cell, we can have totipotent, pluripotent, multipotent, or unipotent. And the difference between each of those levels is how much that particular stem cell can be differentiated. For example, if we take a totipotent stem cell, they are the type of stem cell that can be turned into any other type of cell. They're the most useful, they're the ones, the golden goose as it were. We go to pluripotent. Pluripotent are the ones that more and more are being used in research, and they can differentiate into pretty much every type of cell. Not all, they're not quite as good as totipotent, but certainly they cover the majority of different fields of research and medical treatment. Multipotent are much more limited because they're normally limited down to a particular group of cells. So for example, a blood stem cell would only be differentiated into different types of cells linked to blood. And then of course we have unipotent, and unipotent is where we end up that that particular stem cell can only turn into one particular type of cell. Now there's a big question, should embryo research be allowed? And it is one that has been going on for quite a considerable amount of time. In order to answer that question, I'm going to refer you to a TED talk by Susan Solomon, which is The Promise of Research with Stem Cells. If you type that into Google, you'll be able to find the TED Talk on the TED website, and it's very, very, very informative, folks, so please do take the time to go and watch that. I mentioned earlier that pluripotent cells are the most common and most useful of the stem cells in terms of research, specifically because the other type, the totipotent cells, often result in the destruction of a zygote, which obviously comes with its own ethical issues. The pluripotent cells are quite diverse in what we can turn them into, but they're also quite capable of being produced through a reverse engineering of multipotent cells. So we can actually take a multipotent cell and we can do some magic jiggery pokery. You don't need to know the details of how, but we can then turn it into a pluripotent cell. By turning it into a pluripotent stem cell, we then have the ability to turn that stem cell into a different type of cell. Once we've done that, we can then use that for a range of different treatments, up to and including treatments of things like Parkinson's disease and Alzheimer's disease and a lot of these more recent neurological diseases that you see hitting the headlines. What you've got in front of you is a list of different types of stem cell locations the stem cells can be found, and a list of diseases that they could potentially be used or even are being used to treat. It's very unlikely that they're going to ask you a very specific question from memory in the exam, but they could give you information about a particular location that cells have been sampled from and ask you to come up with what kind of things this may be used to treat. So it's a little bit of common knowledge. Pause the presentation, try and match up the type of cell, the particular disease, and when you're ready you can restart the presentation and go through the answers. So pause now. And let's have a look at some answers. So again, you just have to come up with the connection between the disease and the particular part of the body that is affected by that disease. Once you know the particular part of the body that's affected by the disease, that is the type of stem cell or location of stem cell that you're going to want to use in treatment for that particular disease. As we've said, there's a number of different things we've looked at. We talked about totipotent cells. Remember, these are the ones that we got from an embryo. We talked about how we can get totipotent and totipotent cells when they are differentiated, they become specialized. Again, you need to be able to use the terms differentiate and specialized. We needed to talk about where we can get stem cells from and the different types of stem cells in terms of unipotent, multipotent, pluripotent, and totipotent. You also should have some idea about the treatment of humans and how the different pluripotent cells can be used to treat specific diseases. Just to finish off, for each of the points that you've just covered that you should have gone through in the presentation, go through, pick up a maximum of 5 words and try and link 5 words to each of those points to give you an answer